For decades, scientists have been stumped by one of humanity's greatest genetic mysteries, a blood type that shouldn't exist according to evolution, a mutation so rare and concentrated in specific populations that it sparked theories about alien origins and lost civilizations. But in 2025, DNA finally revealed the truth, and it's far more fascinating than anyone imagined. The story of Rh-negative blood begins in 1940, with a routine blood test that would unravel one of the most compelling puzzles in human genetics. Scientists discovered that approximately 15% of people were missing a critical protein on the surface of their red blood cells. This protein, found in all primates and all other human populations, should have been universal, but it wasn't, and for nearly a century nobody could explain why. The protein in question is called the rhesus factor, named after the rhesus monkey where it was first identified. When you have it, you're classified as Rh positive. When you don't, you're Rh negative. On the surface, this seems like a minor distinction, a simple variation in human biology. But the deeper scientists looked, the more mysterious it became. Here was a trait that violated the fundamental expectations of evolutionary biology. If all humans descended from the same ancestors, how could some people lack a protein that everyone else had? And if this was truly an evolutionary mistake, why did it persist in certain populations at such high frequencies? The mystery deepened when researchers noticed something even more striking. Rh-negative blood wasn't randomly distributed across the globe. It appeared with peculiar concentration in specific regions. In most of Asia, Africa, and the Americas, it remained vanishingly rare, appearing in less than 1% of populations. But travel to Europe, particularly to northwestern Europe, and the frequency jumped to 15 or 17%. Travel to the Basque region of Spain and France, tucked into the Western Pyrenees Mountains, and you'd find the highest concentration anywhere on Earth. Between 30 and 35% of Basque people carry Rh-negative blood. Some genetic studies put the allele frequency even higher, at nearly 47%. For centuries, this anomaly spawned wild theories. Some researchers proposed that Rh-negative individuals were descendants of an ancient advanced civilization like Atlantis. Others, taking an even more speculative turn, suggested that Rh-negative blood represented genetic contribution from extraterrestrial beings who had interbred with early humans. These ideas captured the imagination of millions, spreading through books, documentaries, and now through social media and video platforms. The mystery seemed to demand an exotic explanation. But science doesn't work through speculation alone. It requires evidence, rigorous testing, and the kind of methodical investigation that finally came to fruition in the early 2020s. When researchers could finally unlock the genetic code behind Rh-negative blood, they discovered something remarkable. The answer wasn't alien, it wasn't supernatural. But it was, in many ways, far more beautiful than any fiction could suggest. The breakthrough came through advances in genetic sequencing technology and the accumulation of DNA data from diverse populations around the world. Scientists studying the RHD gene, which codes for the rhesus protein, found something extraordinary. The Rh-negative phenotype, the observable characteristic of lacking the rhesus protein, resulted from a complete deletion of the RHD gene itself. This wasn't a mutation in the traditional sense. It was as if someone had taken scissors to the genetic code and removed an entire section of DNA. Understanding how this deletion occurred required diving into the intricate mechanics of genetic recombination. The RHD gene sits between two nearly identical DNA segments called rhesus boxes. These boxes share such remarkable similarity, approximately 98.6% homology, that during the process of meiosis, when cells divide to create sperm and egg, the genetic machinery can become confused. When DNA strands line up to exchange information, these identical rhesus boxes can misalign. Instead of matching up perfectly, one strand pairs with a shifted position on its partner. When the strands separate after this unequal crossover, one chromosome ends up lacking the RHD gene entirely, while the other receives a duplication. This process, occurring over thousands of generations, created what scientists call the hybrid rhesus box, a telltale genetic signature marking where the deletion occurred. This mechanism, while fascinating, raises an immediate question. If this deletion could happen to anyone, anywhere, why does it concentrate so dramatically in European populations? Why are the Basque people such extreme outliers? The answer lies not in the mutation itself, but in the history of human populations after the mutation emerged. According to the latest genetic research published in 2025 and 2026, the Rh-negative allele likely originated roughly 30,000 to 35,000 years ago, somewhere in the ancestral human populations of Europe or Western Asia. At that time, anatomically modern humans had spread out of Africa, but were still adapting to their new environments in Eurasia. The mutation appeared, probably in a single individual or small family group, and for reasons not entirely clear, it didn't disappear. Instead, It persisted, it spread, and in certain isolated populations, it increased to remarkable frequencies. The key to understanding this concentration lies in ancient DNA. In recent years, paleogenomicists have extracted and sequenced DNA from ancient human remains, allowing them to track population movements and genetic changes across thousands of years. What they found revealed a crucial truth about modern European genetics. The Rh-negative allele correlates strongly with ancestry from Western hunter-gatherers, or WHG, as they're abbreviated in scientific literature. These were the people who inhabited Europe before the arrival of Neolithic farmers from the Near East and later steppe pastoralists from Central Asia. Genetic modeling suggests that Western hunter-gatherers carried the Rh-negative allele at approximately 24% frequency. Neolithic farmers from the Near East carried it at only about 4%. Steppe pastoralists added their own genetic contribution, but the critical point is this. In populations that maintained high Western hunter-gatherer ancestry, the Rh-negative frequency remained elevated. And nowhere is this more true than among the Basque people. The Basques are among the most genetically isolated populations in Europe, something reflected in their unique language, Euskara, which has no known relationship to any other European language and may represent one of Europe's oldest linguistic traditions. Their isolation wasn't accidental. The Basques inhabited the Pyrenees Mountains, a geographic barrier that made their region relatively inaccessible to the population movements that reshaped much of Europe. When Indo-European peoples swept across the continent around 5,000 years ago, when Roman legions marched through most of Europe, when medieval kingdoms rose and fell, the Basque people remained in their mountain stronghold, relatively isolated from outside genetic influence. This isolation had a profound effect. The Basque population preserved ancient European genetic signatures to a degree found nowhere else on the continent. They maintained higher frequencies of Western hunter-gatherer ancestry than virtually any other European group, and consequently, they maintained higher frequencies of the Rh-negative allele. It's a remarkable example of genetic continuity, a population that has maintained remarkable genetic similarity over thousands of years, not because of some exotic origin, but simply because geography limited the amount of external genetic mixing. Recent genomic studies have nuanced this picture slightly. A comprehensive 2021 genome-wide study found that Basques are clearly differentiated from surrounding populations, but not because they come from some wholly separate external origin. Instead, their distinctiveness reflects genetic continuity from the Iron Age onward, combined with relatively limited gene flow from outside populations. Interestingly, the 2018 RH study found that Basques sit at the high end of a distribution, with similarly elevated frequencies in nearby North Iberian populations, suggesting a regional gradient rather than a uniquely Basque phenomenon. But the Basque region still represents the apex of this distribution, the place on Earth where Rh-negative blood reaches its highest natural frequency. Yet this explanation, while scientifically sound and supported by extensive genetic evidence, represents only part of the story. The existence of Rh-negative blood in European populations raised another puzzle for evolutionary biologists. Why would this mutation persist at such high frequencies when, prior to modern medical intervention, it carried significant fitness costs? The answer connects to one of the most tragic medical complications of pregnancy. When an Rh-negative mother carries an Rh-positive fetus, incompatibility can occur. During pregnancy, and especially during delivery, fetal blood can enter the maternal circulation. If this happens, the mother's immune system, recognizing the fetal blood as foreign due to the Rh-positive antigen, can produce anti-D antibodies. In the first pregnancy, this typically doesn't cause serious problems, but in subsequent pregnancies, these antibodies can cross the placenta and attack the fetal red blood cells, causing hemolytic disease of the newborn, a condition that could cause severe anemia, brain damage, or even fetal death. Before modern medicine, this was a genuine threat to reproductive success. Rh-negative women with Rh-positive partners had elevated risk of losing children to this condition. Yet the Rh-negative allele didn't disappear. This shouldn't happen according to basic evolutionary theory. Selection pressure should have eliminated such a deleterious trait. This puzzle led scientists to propose an intriguing hypothesis. Perhaps the Rh-negative allele carried some selective advantage that offset its reproductive costs. Perhaps it conveyed benefits that made up for the risk it posed during pregnancy. Recent paleogenomic evidence suggests one possibility. A 2025 study found that Western hunter-gatherer ancestry, of which the Rh-negative allele is a marker, correlates with extreme longevity in modern populations. Could it be that Rh-negative carriers, or more accurately, carriers of the broader set of genetic variants associated with Western hunter-gatherer ancestry, experienced longer lifespans that compensated for reduced fertility? This would explain why the allele persisted at such high frequencies, despite its reproductive costs. The researchers who proposed this model described it as synergistic selection, multiple genetic advantages working together to maintain a trait that, viewed in isolation, seemed disadvantageous. This hypothesis remains speculative, requiring further research to fully test, but it illustrates how modern genetics approaches these ancient puzzles, searching for mechanisms that can explain the patterns we observe rather than defaulting to exotic explanations. Another crucial finding from DNA research concerns what Rh-negative blood is not. For decades, some theorists proposed Neanderthal heritage as an explanation. If Rh-negative humans had interbred with Neanderthals, perhaps this blood type represented a genetic contribution from our extinct cousins. This theory had a superficial appeal. It provided an alternative explanation for an anomalous trait, but DNA evidence definitively closed this door. When paleogeneticists extracted and sequenced Neanderthal DNA from ancient remains, particularly from the Denisova and Altai Neanderthal specimens, they made an unambiguous finding. Both specimens were homozygous for the ancestral A variant at the chromosome position that determines rhesus type in modern humans. Both Neanderthals were Rh positive, not just phenotypically, but at the genetic level, they carried no trace of the Rh-negative allele. Moreover, comparison of Neanderthal Rh gene variants to modern humans showed that Neanderthals possessed Rh variants that were compatible with each other, maintaining stable genetic patterns for approximately 80,000 years. This finding is significant. It demonstrates that Rh-negative blood didn't come from Neanderthal introgression. The mutations that created Rh-negative blood emerged independently in anatomically modern humans, after our species had diverged from our Neanderthal cousins. This is actually more remarkable in some ways. It shows that the same selective pressures that shaped modern human genetics, pressures related to environment, diet, disease, and reproduction, could generate independent variants in different human groups. The story of Rh-negative blood research also reveals something important about how science actually works and how scientific understanding evolves. For nearly a century after its discovery, the Rh blood group system remained mysterious. Researchers knew the basic facts. They knew it was genetic, they knew about the population distribution, but they couldn't explain the mechanism. The limitation wasn't scientific curiosity or effort. It was technological capability. Understanding what the Rh-negative mutation actually was required molecular biological techniques that simply didn't exist before the 1990s and early 2000s. Scientists needed the ability to sequence DNA, to identify specific genes, to compare genetic sequences across populations, and to trace genetic ancestry through ancient DNA. As these technologies developed, answers emerged. The hybrid rhesus box was identified. The mechanism of unequal crossover was documented. Population genetic modeling became possible. And gradually, the mystery transformed into understood science. This progression illustrates a crucial point about scientific knowledge. When we encounter mysteries that seem to resist explanation, the solution doesn't necessarily lie in exotic theories. Often, the answer lies in the future development of better tools to investigate the problem. The mystery of Rh-negative blood didn't require invoking aliens or lost civilizations. It required patience, technological advancement, and the methodical accumulation of evidence. The clinical implications of these genetic discoveries continue to matter enormously. Understanding the molecular basis of Rh-negative blood has practical applications in transfusion medicine and obstetrics. Hospitals worldwide must maintain supplies of Rh-negative blood for emergencies, as Rh-negative individuals can only receive Rh-negative transfusions without risking an immune reaction. Pregnant Rh-negative women now receive prophylaxis, injections of anti-D immunoglobulin, to prevent sensitization if their fetus is Rh positive. This treatment, developed in the 1960s and now administered worldwide, has prevented countless cases of hemolytic disease of the newborn. But the treatment is only possible because medical professionals understand the underlying immunology. Recent molecular genetic research has refined this understanding even further. Scientists have identified not just complete RHD deletions, but partial deletions and variant forms of the RHD gene that produce weak expression of the D antigen. Some populations carry a variant called Asian DEL, where individuals are serologically D negative but can safely receive D positive blood without developing alloimmunization. These discoveries have practical implications for blood bank management and transfusion protocols. The distribution of Rh-negative blood also tells us something important about human history. The concentration in European populations, and particularly in Western European populations with stronger Western hunter-gatherer ancestry, reflects deep historical patterns. These are populations that have maintained relatively high genetic continuity with the hunter-gatherers who inhabited Europe before the agricultural revolution. They represent a genetic window into the pre-agricultural past of Europe. By contrast, the near absence of Rh-negative blood in sub-Saharan Africa reflects the fact that most African populations descend from more recent common ancestors, and the African continent didn't experience the same major population replacements that occurred in Europe. In East Asia and most of the Americas, the low frequency of Rh-negative blood similarly reflects different ancestral population compositions and different evolutionary pathways. Understanding these patterns requires understanding what geneticists call demographic history. This is the study of how populations moved, merged, separated, and replaced each other over time. It's the recognition that human genetics isn't uniform across the planet. Different regions have different evolutionary histories, and those histories are written in our DNA, visible to researchers with the tools to read them. One of the most interesting aspects of Rh-negative research concerns the Basque case specifically. The Basque people have long fascinated anthropologists and geneticists precisely because they're so genetically distinctive. Their language, Euskara, belongs to no known language family. Their genetics show patterns unlike other European populations. Their cultural practices and traditions differ markedly from surrounding groups. Yet they're not exotic in origin. They're not a mysterious population that arrived from somewhere else. They're descendants of early European populations who simply maintained their genetic and cultural distinctiveness through isolation. This is actually a common pattern in human genetics. Isolated populations often show greater genetic distinctiveness, not because they have exotic origins, but because they lack the genetic mixing that homogenizes larger populations. The Basques represent an extreme case, but similar patterns appear in other isolated groups around the world. Indigenous populations on remote islands often show unusual genetic signatures. Mountain populations in various regions maintain distinctive genetic profiles. These aren't necessarily markers of exotic origins. They're often simply markers of isolation. The story of Rh-negative blood also illuminates something about how popular narratives develop around scientific mysteries. For decades, Rh-negative blood was described as mysterious and unexplained. Documentaries were made, books were written, online communities formed around theories about its origins. The lack of explanation created space for speculation, and speculation filled that space with increasingly exotic proposals. But this dynamic changed once the scientific explanation emerged. Once researchers could point to the hybrid rhesus box, could explain the unequal crossover mechanism, could present the paleogenomic evidence linking Rh-negative frequencies to Western hunter-gatherer ancestry, the exotic theories lost their explanatory power. They couldn't compete with an actual evidence-based mechanism that explained the observations. This is how science typically progresses. A mystery captures imagination. Speculative theories proliferate. Then evidence emerges, understanding develops, and the mystery transforms into mundane science. What was once inexplicable becomes routine knowledge in textbooks. The excitement doesn't necessarily disappear, but it shifts. Instead of excitement about the mystery itself, the excitement comes from understanding how nature actually works, from recognizing how elegantly evolution and genetics produce the patterns we observe. The future of Rh-negative blood research will likely involve even finer resolution. Scientists continue to sequence more ancient genomes, revealing the genetic composition of populations from thousands of years ago. This should allow increasingly precise modeling of when and where the Rh-negative allele emerged, and how it spread through different populations. More detailed studies of Rh-negative populations in regions previously understudied, parts of Asia, Africa, and the Americas, should reveal whether Rh-negative blood emerged only once or multiple times through parallel mutations. Studies of the selective advantages, if any, conferred by Rh-negative ancestry should clarify whether the hypothesis about Western hunter-gatherer longevity holds up to scrutiny. There are also practical applications on the horizon. As reproductive genetic technologies advance, understanding of HD genetics may lead to better prediction of Rh incompatibility risk in pregnancy. Increasingly personalized medicine approaches might even eventually allow matching transfusions based on more detailed genetic and immunological profiles. But perhaps the most important takeaway from the story of Rh-negative blood is a reminder about how science works. Mysteries that seem profound and inexplicable often yield to patient investigation and advancing technology. Explanations that invoke the exotic, whether aliens, lost civilizations, or other speculative theories, often feel more satisfying than mundane scientific explanations. But the mundane explanations have the advantage of actually being true. They're grounded in evidence. They can be tested. They can be refined and improved as new data emerges. The origin of Rh-negative blood was never as simple as popular narratives suggested. It wasn't a simple mystery waiting for a single, simple answer. Instead, it was a genuinely complex genetic phenomenon, resulting from mutations, population movements, selective pressures, and thousands of years of human history. Understanding it required advances in molecular biology, paleogenomics, population genetics, and evolutionary theory. It required data from diverse populations around the world, ancient DNA from archaeological remains, and sophisticated computational modeling. And in the end, the actual answer, a genetic deletion in a population that happened to maintain high ancestry from Western hunter-gatherers due to geographic isolation, is perhaps more fascinating than any fiction could be. It's a story written in our DNA, a narrative of human evolution, migration, and adaptation, stretching back 35,000 years. It's the story of how we got here, encoded in the very cells that make us who we are. And unlike alien theories or mythical civilizations, it's a story that's actually true, grounded in evidence and waiting to be discovered by anyone willing to look into the science.